Okay, welcome everybody to our collab session. This is presented by Wasatch BioLabs, and it's entitled From Arrays to Sequencing, Enhancing Methylation Analysis for Biomarker Discovery and Clinical Applications. My name is Kathleen Barnes. I'm a Senior Vice President of Population Health and Precision Medicine at Oxford Nanopore Technologies. And it's just really exciting to be here today because we just made an announcement a few days ago about our collaboration between Oxford Nanopore and the Wasatch BioLabs folks. And this is to develop a direct whole methylome sequencing product. And we're confident that this is going to drive innovation in genomic and epigenomic analysis and really accelerate breakthroughs in research and ultimately clinical applications. So to learn more about this new platform, I have a real pleasure of introducing our two panelists. So Dr. Jonathan Hill, who's VP of Science and Technology at Wasatch Labs, and Dr. Tim Jenkins, who's VP of Research and Development at Wasatch. So the way we're going to navigate this 30-minute session is Tim and Jonathan are going to tag team in a brief overview slide presentation, and then we're going to jump into questions over here. I'll ask a few questions to start, and then we'll open it up to the audience. So without further ado, I think, Tim, you're first, so we'll let you go.
It's really great to be here with everybody and excited to have a chance to talk a little bit about our company that we've built out of a desire to try to actually impact the clinic with epigenetic-derived diagnostic tests. This is something that I've focused on for a large portion of my career and have found a lot of roadblocks along the way. So we set out to build this company specifically to identify and to prioritize clinical actionability with diagnostic test generation. We've done this on our own with development of a handful of diagnostic tests. And as we have gone through the process, we've recognized that we needed to almost build our own pipeline and platform for getting these to a point where they could actually be used in a clinical setting and pass all the barriers associated with CLIA and FDA. So our goal is to develop some of these tests on our own. But in addition, we are excited about the opportunity to take this same pipeline that we've built and almost crowdsource it. We know lots and lots of people are working on some of these same ideas in lots of different for lots of different indications, and so we believe that by working and partnering with other folks, we can really realize the potential that exists in epigenetic diagnostics. Everything we do is based on four basic principles. The tests we generate have to be simple and direct. They have to be scalable, they have to be highly reproducible, and they need to be data rich. When we first set out to accomplish the goal that I've described before, we realized that the vast majority of technologies that are available to us fall short in one of multiple ways. Often we have issues with limited coverage, issues especially with having to manipulate the DNA to a large degree to actually be able to do any sort of analysis, which creates all sorts of biases. And then big issues with batch effects. And we set out to identify technologies or a platform upon which we could build some of the tools that we were looking at, and we eventually landed on Oxford Nanopore. The reason for this is because it overcomes these major limitations. We get comprehensive coverage, minimal manipulation of the DNA, and it's highly reproducible, and I'll show you just how highly reproducible it is here in just a minute. But using this technology as kind of a base upon which we build all of our tools has really given us a lot of advantages, and I hope to show you that here in just a second. One of the issues, though, when we started working with Oxford Nanopore, I don't think it's any secret that Oxford Nanopore did not start necessarily to be directly implemented in the clinic, right? So we realized early on that if we were going to utilize this technology, we almost had to build platforms and build tools to kind of plug into the Oxford Nanopore platform itself. And so we've generated what we call our kind of path to the clinic, where we have early stage testing that allows us to assess kind of biomarker discovery in the first stage, which includes everything from kind of a modified version of of what Oxford Nanopore already offers, in addition to a very specific data analytical pathway that simplifies everything and makes this very easy to use. And we believe that using these techniques, we can then turn the corner and get ourselves into clinical deployment. We're in fact building out a 20,000 square foot lab right now in an effort to, again, move our own tests forward, but to also partner with many other folks to move these tests forward in an environment that can be FDA regulated so that it has the legs to move forward and have clinical efficacy. I will just focus on the beginning of this pipeline, by the way, here, just so that we're all aware, as Jonathan and I will both focus on this. The kind of the key step for discovery is direct whole methylome sequencing is what we're calling it. Effectively, we won't go into all the details, but it involves a proprietary shearing protocol that's very inexpensive and can be used kind of in a centralized fashion, something that's sent to us, or something decentralized that's off in your own laboratories. We use a modified nanopore, a typical nanopore prep, but modified in a couple different ways to ensure that we can have that we can do multiplexing appropriately to make this as inexpensive as possible. We do direct sequencing on a PromethION flow cell, and we've generated the MEthylSeeker software package, which is a really, really important piece of the puzzle that allows this really complex data to be very, very easily analyzed. Just to show you exactly how reproducible dWMS has proven to be, we did an assessment with the exact same tissue, blood, from 4 individuals, and we did it on both epigenetic and we did it on dWMS. We sent this additionally to 2 different locations. Different location, different machine, different lab technician, all of the things that we need to worry about with batch effects. With array, we found that the number one cause of variance between those 2 samples were the location where it was ran. Somewhere between 20 and 35% of the variance that exists has everything to do with which batch it was running, which makes it incredibly difficult for you to actually use a clinically actionable test. Interestingly, when we used dWMS, we were unable to find any principal component or any variance associated with the location in which it was run. And so we can dive into more details if there's more interest there. But this is an incredibly important thing for us to be able to move something forward into the clinical space. It's also important to note that we needed to check to see exactly how much data we needed to perform a clinical diagnostic test. And we found that at about 5x coverage, we can get 97, 98% of all CPG islands that we were interested in. And because of that, we didn't need to necessarily go a whole lot deeper, and that's where we started digging. You can see on this figure here that this line right here represents CPG islands that have 5 or more probes on the EPIC array, 3 or more probes, and 1 or more probes, 3 or more probes, or 5 or more probes present on that EPIC array. We also thought it important to see if we would get the same information from dWMS than you would with array. And we performed a very simple analysis where we looked between 2 distinct tissues. The exact same samples were run on both dWMS and array. And this is dWMS at only 5x coverage. And we found that virtually all of the positions that were identified with array, we were also able to identify, but that dWMS identified far more that were not identified by array. And these are just CPG islands. We tried to set it up so that it was a fair fight. The data speaks for itself. Importantly, it's not just CPG islands that we're interested in looking at either. It's also the fact that we can have so much more data available to us. We can have data that is associated read by read, and we can determine which cell or which cell type each piece of DNA actually comes from. This was a simple analysis done with various neuron cell types and blood to demonstrate that this is a possibility, and this is over only 100 base pair window that we're able to identify this much. The biggest issue that we faced growing this out, though, is now we have this incredibly rich data, and we have to figure out a way to analyze it. I'll hand it over to Jonathan, who was the key in developing the MEthylSeeker package, to talk about how that works.
Thank you, Tim. As he mentioned, we get far more data using dWMS than we're used to working with methyl array data. And this means that we have to consider things that we've never had to worry about just because of the limitations of our computers that we're using. And this kind of can break down into two major parts, right? First is just simply storing all of that data and transferring that data. To give you an idea of the scale of difference here, on an array you typically have a 900,000 spot array with that many CPGs. We on average get 27.5 million CPGs that are covered in our data, and we get multiple calls per CPG. And so on average, we have 30 to 50 times the amount of data that we're used to working with. So, storing that data becomes an issue as you build up sample numbers, et cetera, and transferring it. If you need to upload it to the cloud or download it from a service provider, these become real issues. The other one is simply analyzing it. Turns out that R, for example, can have 2.8 billion items in a list. That's its limit. We regularly get beyond that limit, and R crashes trying to analyze this data. And so we have created two solutions to both of these problems. The first one I'm going to talk about is a custom file format that we've made to aid the storage and transfer of our data, and then a methyl-seeker R package that will help us be able to analyze and manage this data even when it's too big for the memory of our computer. So first, just to go into the format, we call this the CH3 format, named after the chemical formula for a methylation mark. What we do is we take the nanopore methylation calls directly from their pipeline. We combine it with the Parquet file format, which is an open standard with APIs in every programming language that allows us to efficiently store this. And then we've created a custom schema to sort and select the data that we're storing to maximize efficiency. The bottom line is we take what was a tab delimited file that on average at 5x coverage was about 20 gigabytes, and we can compress that down to 1 gigabyte per sample. And so of course when you're talking 10, 20, or 100 samples, this just magnifies the difference in your data storage and efficiency. And interestingly, this compact file format actually reads faster and performs better than a tab delimited file does as well. Now we have to be able to use that file, and so we've created the methyl-seeker package. Currently is made in R, but it actually could be adapted to Python or any other language because the key components exist in all languages. We take that CH3 file, we import it into an on-disk database based on the DuckDB framework, and that creates our initial kind of position read summaries. Then we can do different analyses with it. We can summarize by sliding windows across the genome. And as it does so, it does it in batches and writes it back to the database so we can manage our memory. We can also do it by predefined regions such as CPG islands. And in both cases, so really at the end here, you can take the position data, the Windows data, or the regions data and calculate your differential methylation. The functions exist for that within this package. And what's really cool is what it ends up being is that in 3 commands of code, load up the database, summarize however you want, and then calculate your differential methylation, you can run your complete analysis. And all of the database management and behind the scenes stuff that most of us biologists don't want to deal with is taken care of by the package. You don't have to worry about any of it. If you come back and analyze it later, it's saved on disk, so you don't have to rerun your pipelines either. You can pick up where you left off. So, of course, that enables all the kinds of analyses you would typically do. This is some of that cell type analysis that we showed earlier, but I just want to give you a flavor of some of the plotting and analyses that you can build on top of this package. And of course, software packages are cool because as we go forward, we will expand its capabilities and incorporate other things. We're working right now to incorporate it directly into nanopore-based pipelines, as well as to add machine learning and regression analysis functionality to this package. So just to kind of bring it all back together and recap what we've talked about, through the ONT platform and then what we've built on top of that, we have finally created a technique or a pathway that checks all the boxes that you need for something to be able to get to the clinic. And we at Wasatch BioLabs are very interested in this crowdsourcing model. We don't have the specific disease knowledge, et cetera, that many of you have. So we want to partner with you to take things from the lab, work it through a common platform that can be the same platform for discovery as it is in the clinic, and help you each step along the way to be able to get that to where it's impacting patients' lives. So with that, we'll finish up. If you have some more, if you're interested in more information, we have a case study that you can look at dWMS information. We also have people at booth 404 who are happy to talk to you about it and what potential there is for partnership. So with that, I'll turn it back over to Kathleen.
Thank you so much, Tim and Jonathan. And before I open it up, I have a couple questions to ask the two of you. First of all, thanks so much. This is so exciting for those of us who have been in the multi-omic world of translational research. We've tended to think of, whole methylome in applications like epigenetic, genome-wide applications, QTL. There's been an explosion over the last few years, I would say since about 2018, in methylation risk scores and the potential power of methylation risk scores in clinical applications. This morning, in anticipation of this presentation, I just did a quick PubMed search on the term methylation risk score. There are 10,444 publications in peer-reviewed journals. And so my first question, I think I'll direct it to you, Tim. Based on this, given the limited tools that we've had in our toolbox for this technology, how do you see direct whole methylome sequencing enabling the broader expansion, and most importantly, moving it to the clinical arena?
Yeah, so this is something that literally throughout my whole career we've been attempting to do. Most of them have been array-based tests and methylation risk scores that we've built. We kept identifying issues with batch effects. Quite literally, we've built 2 different tests that never quite saw the light of day because we couldn't get past some of those major issues. Also, with the dWMS platform, it really takes you from the front to the back. So using the exact same technology, you can discover the sites that you need to be using for your risk score calculation or whatever it is, cell of origin calculation. You can use that same technique all the way through, and it can be placed in a what we're building is this 20,000 square foot lab that will help from the development of the test to regulatory and compliance steps, right? And so you can use it all the way throughout, but we're also developing technologies that allow for targeting specific regions, right? So right now we can multiplex these frequently at 4 per flow cell, and likely that can be expanded out with different iterations. But if you start targeting in a really efficient way, we can do that at a much larger scale.
Sure.
Cost goes down. It makes it very, very tangible for lots of people.
Can you tell us a little bit more about your goals to facilitate and enable researchers using the ONT platform, in addition to the big clinical goals that we were just discussing, both as a centralized model but also as a decentralized model?
Yeah, so I think one of our big foci is just to remove barriers, right, for people, whatever research they're doing. And that includes basic research as well. We just recently went through and actually sequenced and created the methylome for 2 different sea sponge species with a collaborator, right? We have so much power where now you don't need to be limited to the couple species that have arrays ready for them. You can do it in any species, including species that don't even have a genome, because you get the sequence data at the same time. So that's one level. And then as we move through this clinical validation, it's all about making it so that we can have a common platform that we work on so that we can overcome those barriers and move through those. And you mentioned the distributed and centralized. We're also very flexible on how this works. Many people don't want to deal with learning a new sequencing technology or setting up equipment in their lab. Send your samples to us. We can run them in our 20,000 square foot space, and we can help you with data analysis. We can help you with any of that you want. However, if you are someone that's already using Nanopore, you already have things in the lab, maybe you already have a set of biomarkers that you validated, you still have to find someone that can go through the clinical validation, regulatory, and then servicing the actual product, right? And we're happy to bring people in at that point and work with them on those steps as well. So it's all about what do we need to get to the clinic and how can we help you get there?
That's great. I am going to, can I pass that to you? I'm going to open this up to the audience. We have a few more minutes. If anyone has questions.
I'm very curious to learn about the input that you need for this test, because EPIC array requires quite some material to analyze.
So Sorry, could you say it one more time?
Yeah, so for epic array, you would require quite some amount of material, meaning hundreds of nanograms of DNA. What about your protocol?
Yeah, that's a great question. 500 nanograms is approximately like minimum. It's nice to have up to a microgram. That has more to do with the shearing protocol, just making sure that is done efficiently and you don't have to worry too much about lowering the volume again. But yeah, it's same range. I wouldn't say that it would go down to like 100 or 50 nanograms, but 200 to 500, like it would be with array, is a reasonable range. Yeah.
Great, thank you.
Yep.
Hi, can you comment on the storage and shipping logistics for samples for the high molecular weight DNA that you need?
Yeah, so interestingly, you actually don't need high molecular weight DNA if you don't want to for this application. So extraction with like a D&EZ kit, something like that, is totally fine. And it would just need to be on ice in shipment. We've actually done some work recently on the thermostability of methylation marks. It's something we'll publish here in the near future. And it is important that once extracted, that it is on ice. But other than that, it doesn't need to be in a dewar or on dry ice or anything like that. Yep.
Hi, great talk. So I have a question about the reproducibility study you did. In what sample types have you demonstrated the reproducibility? For example, did you do anything in oral samples like saliva or buccal swabs?
So we haven't done. Correct me if I'm wrong. I don't think we've sequenced buccal swabs. I don't think we've done reproducibility study directly on buccal swabs. We've done it on. I'm a reproductive biologist, a male reproductive biologist by trade, and so we did it. In fact, what you looked at there was tissue, the sperm tissue and blood, and so those were the two that we've done it on. We have sequenced mouse, zebrafish, sea sponge. We've done buccal swabs. We've done a number of different things and never seen issues at all. In every time that we have included a batch effect analysis, it doesn't exist. I can tell you. Let me tell you more scientifically. We were able to go down to like a PC8, I think is what we did, principal component number 8, and it got down to 10 to the negative 28, but that still wasn't the batch. So we've never seen the batch. It's definitely less than 10 to the negative 28%. But at that point, it doesn't really matter, obviously. So yeah.
I have a follow-up question on like the multiplexability and pricing. Are you allowed to share like what's the multiplexability for your platform and what would be the cost comparing to the EPIC array, for example?
Yeah. So cost is still being worked out. Multiplexing right now will be probably at 4 up to 6. It also depends. Like this is also for the discovery phase. Once you get into targeted phases, we're talking 24, 48 per flow cell, which becomes very, very reasonable quickly. So yeah, very, very competitive with what else is on the market.
Thank you.
Yep.
Hi, my name is Varun from True Diagnostic. Great presentation, by the way. This is exciting technology. One of the things that we do is that we use epic arrays to do a lot of like age calculations, methylation risk scores. So I really appreciate bringing that up, that you brought that up. One of the things about these algorithms are that the weights that are part of these epigenetic clocks, if you will, are optimized for the epic array. How are you guys thinking about doing taking your platform and maybe making it viable for something like Dunedin Pace or the Grim Age clocks or some that have been developed before? So
I actually made the first, because it turns out like Horvath's clock doesn't work for sperm, and that was the area of interest that I had. So I actually was one of the first to build a sperm epigenetic clock. The fascinating thing about this, and we built it on array, the biggest problem that we had is that for every single batch, we had to normalize and move it. We had to move that number back and forth because the batches were problematic. I've actually started to do this with some of the sperm data that we have to track aging signals. They're effectively they're all in the same regions. We regionalize all of our data typically when it's coming off. I know Horvath's uses single point data, but it's frankly, in all of the assays I've done, it's much better regionally. So we regionalize, and then we're sitting at the same positions, we're sitting at the same regions. It might take a small study to retrain on this new technique, but I would argue that it will be more stable. and there will be less correction at the end. All of those things are not going to be as big of an issue. So that would be, yeah.
Thank you.
Hopefully
No,
that's
that's
helpful.
great. And actually, I read the paper, so I didn't realize that was you.
Oh, yeah.
Nice to meet you.
There you go. Yeah, nice to meet you too, for sure.
If there are no other questions from the audience, I'll ask you guys one last question. We've talked a lot about application around methylation risk score. I'm passionate about it. But can the two of you speculate for us, just in closing, other potential applications? Jonathan, you've thought a lot about building classifiers using these data that would be so much more accurate. Can you guys comment on that a little bit?
Yeah, so I think, what can we do with the methylation data when we have it is super important to think about early on, right? What are we going to use this for? We've been using it for kind of two different main types of analyses. One is what we call a tissue of interest classification. What we use for the training data there is actually the tissues of interest, purified cells from those tissues. We get their methylome data, and we find the spots that are uniquely methylated in that tissue of interest. We can then go to something like cell-free DNA in the blood, and we can classify read by read, where did that read come from? And we can tell you your mixtures, right? 10% comes from this tissue, 20% from that tissue, that kind of thing. The other one is more of a traditional methylation risk score, where now we have a large number of patients, and we're doing neural modeling or neural net modeling or GLM net or something like that in order to identify patterns within the population, essentially. Both of them work really, really well. The batch effects help hugely because your training data is never going to be run on the same run as your actual classification data. So you've got to get rid of that batch effect or you're going to mess up your models, and they're very hard to compensate for. And so we've done that. We've been able to figure that out.
Congratulations. Yeah.